There we go. Looks like we are recording. All right. So I'm glad that everyone is here and participating in this. This is our first virtual discussion in quite a while. I think the last one was sometime in 2023. And our topic for today is the optimal host environment for retinal ganglion cell transplantation. So we have kind of a big group of people who are helping facilitate this. And I guess I'm kind of facilitating the facilitators. So, as most of you are aware, the goal of the RReSTORe Consortium is to really look at ways to restore vision in cases of optic neuropathy. And so we have this broad collaborative goal. And really, one of the goals of these virtual discussions is to be able to bring together lots of people from lots of diverse perspectives to share information on the cutting edge of the science, but then also to, through the course of discussion, bring up new areas that might be fruitful for future research or new frontiers that we need to push as a broad, multidisciplinary scientific and clinical community. So today we have some really great expert facilitators who are going to each provide a short introduction on various facets of the optimal host environment for ganglion cell transplantation. And then the goal of that is that we'll kind of seed the discussion so everyone in the group can then participate in this discussion. My request to you is that during the session here, you keep yourself muted until you're ready to jump in and say something. That way we don't pick up kind of background noise, whether it's people knocking on your office door or dogs barking in the background or whatever. And then please chime in and contribute to this good discussion as it takes place. So our facilitators today are Brad Fortune, Jeff Goldberg, Tom Johnson, Peter Baranoff, and Derek Wellesby. They have some really great expertise in this area, and we're excited that they agreed to participate in this session. So I want to start. I'm going to turn this over first to Dr. Brad Fortune, who's going to sort of provide the first set of introductory remarks. So if you want to go ahead and share your screen, and let me stop sharing mine. There we go. And now you can share yours and take it away.
Hi, thank you, Matt. Did that come through? It looks like it's on the
Yep,
share.
everything looks good.
Great. Okay. Thanks very much to Matt and John and all the rest of you for the opportunity to take part and introduce today's discussion. It looks really interesting. And speaking of background noise, I apologize. There's some intermittent, horrific drilling of some sort in the building, and it's coming right through my office, so that'll be in the background, perhaps. So I was asked to, this is the title I was given, Animal Models to Use and the Disease Stage in these models, as well as what a likely human patient might look like. So I took this as an indication to do a bit of an overview or introductory type comments, just to get this out of the way. Some of the images I'll show are from the Spectralis, so we have that disclosure. So in terms of thinking about human patients that might, at least initially in the early trials that might occur in this field, should we get to that stage, uh, along with my collaborators and others in the field, we came to think that most likely the first transplantations would only be done in patients who had no vision to spare. And for that reason, we initially started in our work in this field, and I'll show you what I mean by we, using, in this case, a rather advanced stage of experimental glaucoma in non-human primates, thinking that this would be the most translationally, representative or sensible stage to begin with, that it would mimic the type of patients or eyes in human patients that we, would be seeking to transplant. That said, our group and certainly with influence of others have started thinking more and more about comparing results to transplantations done at earlier stages of experimental glaucoma, including possibly even in relatively naive eyes or completely naive eyes. And so I brought some slides that aren't part of the initial introductory set here, just in case we want to circle back to some of that. That said, I think that development of these approaches, being that this is a relatively new field, are going to require, a full spectrum, is the way I phrased it, of translational models in all different species, the advantages that come from mice, of course, are genetic manipulations, but also the ability to do high throughput at relatively low cost, mice and other rodent models. We do ultimately need larger animal models with eyes that are much more similar to the human being. And there are a host of species that I think fill that gap. So porcine, feline, canine, and simian, which will enable, surgical approaches that would be very, very difficult to achieve in a very small eye like a mouse, um, at least with current instrumentation and optics. It probably also does a better job at predicting graft versus host disease if we're using human cell lines, human-derived cell lines in the translational models. Not all groups are doing that. Some groups are using mouse-derived cell lines to transplant into mice or into tree shrew, for example. There are groups I know at UC Davis working on development of non-human primate or rhesus cell lines to transplant into rhesus. But I also think that another issue is the timelines. So the larger animal models generally have a much slower course of experimental glaucoma than some of the rodent models. And that may be interesting in terms of targeting the stage of damage. You'll need a little bit of dynamic range in both sort of time and progression rate in order to be able to target a given stage. In most of the rodent models, you're at severe end stage damage within a month or so. So let's talk about some of the models. There are, at least a dozen or so inducible models of experimental glaucoma in rodents. I've listed, I think I counted about 8 of them here, just to have a few examples. They seem to mostly be applicable to other species. So, for example, microbeads, steroids, topical steroids, and that's whether it's paramagnetic or latex microbeads. The silicone oil model has been used in non-human primates as well. So almost all of the things that can be done in mice or rats can be done in a non-human primate. Similarly, the tree shrew model offers some advantages. And when I say non-human primates, people have been using rhesus, cynomolgus, squirrel monkeys, marmoset to do experimental glaucoma. There are genetic models, of course, the most well-known being the DBA2J, which actually, follows a course of experimental, it's a high IOP model. The COIP1B1 knockout doesn't exhibit high IOP, but does get an optic neuropathy. And similarly, the myocilin mouse in some labs does not exhibit ocular hypertension, but does get an optic neuropathy and outflow traditional or conventional outflow pathway abnormalities. Interestingly, though, not manifest high IOP. There's the feline mutation. Gillian McClellan's lab, Marcus Kuhn have published extensively on that model of experimental glaucoma. That's a larger eye. Interestingly, among canine, there is a genetic model of basset hounds that have primary angle closure glaucoma. But in a lot of the purebred animals, a lot of purebred dogs, the prevalence of glaucoma is much higher than it is even in people, in human beings. So this stands to reason that genetic sources will be identified increasingly in some of these species. We shouldn't stop at just experimental glaucoma, of course, because ultimately the goal of the RESTORE is to restore vision in all patients with optic neuropathy of varied sources. So Leber's hereditary optic neuropathy, dominant optic atrophy, traumatic optic neuropathy, et cetera. So other models are important to consider, the most well-known of which are probably optic nerve crush or transection, also inducible, can be unilateral. Similarly, traumatic optic neuropathy, either primary or secondary due to traumatic brain injury models. Ischemic, such as the one we worked on many years ago, delivering endothelin to the anterior retroorbital optic nerve via ALSA mini pump. There is an AION model in non-human primates. And of course, NMDA excitotoxicity can be used to kill ganglion cells. Other genetic models include OPA1 mutations, that is a homolog to autosomal dominant optic atrophy in both mice, and recently from the UC Davis group, Sarah Tomasi, Alam Moshiri, and colleagues at UC Baylor have discovered OPA1 mutations in rhesus macaques who develop an optic neuropathy that looks very much like autosomal dominant optic atrophy. And I just want to mention that there are important complementary models that aren't animal models per se, but that involve use of explant. And in this case, I mean retinal explant from mouse or monkey. I think we'll see some results as we go. I brought a few slides from Ben Severe's group. His postdoc Joe Leffler have been doing some really nice work in collaboration with our group in rhesus macaque. We can learn a lot about stem cell engraftment from some of these models, not least how what markers are developing in recently transplanted cells, how their maturation is going in terms of electrophysiological characteristics and morphology, et cetera. I will say one thing, that we gave a pair of retinas, among many others, to Ben Severe and Joe Leffler and colleagues in their lab from an animal that had severe unilateral experimental glaucoma, and the stem cells, the retinal ganglion cells derived from stem cells from Jason Meyer's lab, transplanted onto those explants, performed the same in the two eyes, in the fellow control eye, as it did in the severely glaucomatous eye. So intrinsically, the severely glaucomatous retina doesn't seem to, in and of itself, preclude transplantation. Things went as well in that explant as it did in the fellow control and in many other naive explants as well. So that is interesting, because it then points perhaps to some of the other indications that we've reported at ARVO, for example, and for which I have slides here if we want to circle back, in terms of the stage of damage having a great influence over at least the postoperative success. So what I mean by stage of damage is, I'll just show a few slides here in terms of, this all circles back also to what the human eye might look like in terms of the severe glaucoma stage. So I'm just showing you the way the non-human primate model of experimental glaucoma goes. We use laser photocoagulation and trabecular meshwork to create an outflow infacility and intraocular pressure elevation. Here's a typical example. The laser time points are indicated by the little green diamonds on the timeline here. There's some baseline measurements. And then, as you can see, after, in this particular animal, in this eye anyway, 4 laser photocoagulation sessions resulted in chronic elevation of intraocular pressure. And you can see typical measures derived from OCT scans that are used in a clinical setting all the time. So the optic nerve head minimum rim width measurement, the circumpapillary retinal nerve fiber layer thickness measurement, and the posterior pole ganglion cell complex thickness measurements all decline over time. We tend to be interested in these early time points, the so-called onset. And what I mean by onset in this case, just to define it, is the first observation below test-retest variability. It's actually below the 5th percentile of test-retest variability, as long as it's confirmed. So then you actually go from 95% specificity to 100% specificity if you get a confirmation. MRW onset is almost always before RNFL thickness onset, but here are the SLO images along with some mid-horizontal B scans to show you the increased cupping and excavation, thinning of the rim, progressive thinning of the rim tissue. You can see that here. There's almost none left through this horizontal slice. There's almost none of the inner retina left. We'll go to the next slide and we'll see that you can see the axons by the final time point have completely melted away. None of their reflectivity is left. You can see that along with that has been lost most of the inner retinal capillary plexus. So the radial peripapillary capillaries in this case are all gone. And the flow, blood flow, this is from laser spectrofluorography, the blood flow within the optic nerve head is dramatically reduced as well at that point. So, this is something that we're going to need to contend with both in experimental models as well as in the human condition, that the inner retina is largely gone, that the capillaries that would normally be feeding that tissue are gone. And we have to think about, the ongoing effects of sort of chronic glaucoma. The optic nerve head is, dramatically remodeled. There's not much of a rim tissue left for axons to guide their way into the optic nerve. There will be lots of gliosis, et cetera. So So it's a pretty harsh environment, as if it wasn't harsh enough already with the xenograft from human-derived cells into rhesus macaque. I can stop here. I do have other slides if we want to circle back to any of the questions about how transplant surgery has gone in some of these animals. But just as an overview for the kind of work that we've been doing anyway, and where it sits relative to the work that you'll see, I think, presented by the rest of our speakers today.
Great, thank you. I think I'm going to turn over to Dr. Jeff Goldberg, who's going to give us a few comments on strategies for implantation. So Jeff, if you want to share your slides. Wonderful.
Okay, great. Are we full screen?
Yes, looks good.
Okay, great. Let me just move all the pictures out of the way too. Great. Okay, so Brad has already brought up a couple of like really major points, some of which I'll include, in here, a couple of major points about, disease stage and model selection, and as well as animal selection. And I'm going to, I think those are going to be great points for discussion. Let's see. Okay. So, taking that forward now into neuroprotection and cell replacement, I'll just remind everybody that, donor cells, we talk about donor cells being able to be neuroprotective themselves for the host cells, but also the way that donor cells may be able to modify host environment. Stem cells are sort of broadly known or thought to be anti-inflammatory as an example. And I think that that's going to be a really important element to incorporate into discussion and experimentation now and going forward. A lot of us are focused pretty heavily on mainly putting RGCs into a transplant model, even if they're stem cell derived RGCs, but the other cells, including the stem cells themselves, we and others have published have, profound profound functional implications on the host as well. Obviously, at the end of the day, we are also looking for retinal ganglion cell replacement in these approaches. And today's a great opportunity to really continue this discussion about what's the optimal host environment for transplant and integration. And Matt had also suggested that we try to talk about how much integration is needed for functional restoration. And here, I'm not going to belabor that point other than to say, we have a lot of hand waving and sort of little data hints about what that might look like. But moving a patient from no light perception to light perception is itself going to be an enormous step forward, even if we could just do that. And so thinking about the maybe few thousands of numbers of cells that might be needed in a human one day for that, I think that's going to be a good question too. Brad has already seeded this question about the normal retina versus, the optic neuropathy degenerating retina, whether that's glaucoma or optic nerve crush. We had done some initial work trying to simplify the question and initially just ask about the normal retina. And these are data that we published now 8 years ago, where we showed that donor retinal ganglion cells, primary retinal ganglion cells, purified from one animal and injected into the next animal, in this case, again, with no optic neuropathy model, we got some pretty good evidence of integration in the retina, electrical integration into synaptic layers, so responsiveness to light, shown electrophysiologically. Now, of course, we and many others are doing those kinds of experiments with in vivo calcium imaging and axon growth down the optic nerve, going all the way back to relevant target regions in the brain, not making what look like normal axonal or synaptic distributions there, but at least getting to at least some of the right spots in the brain. So then the follow-up question obviously became, well, what about in the injured retina? And here, Ziming and Kunjer in the lab, Su Chen, who was in the lab, did experiments looking at now, instead of primary RGCs out of one mouse or rat, looking at induced RGCs made in culture out of human stem cells. First of all, if you sort of compare historically, many fewer cells, even into the normal retina, many fewer cells survive and integrate into the retina when we look 1 week or 1 month after transplant. Of course, you can appreciate that even in that time course, the number of surviving cells into the normal retina does go down. So some cells get in but then die. But then really, if we transplant the cells immediately after optic nerve crush, using that as a, very profound injury model, optic neuropathy model, again, you can see many fewer cells successfully integrating. And in these experiments, when Ziming and the group waited to look a month after transplant, maybe it was marginally better, but not statistically significantly better than the 1 week after, or than the transplanting immediately after optic nerve crush. And so, this was looking at like kind of the premise of cell replacement into the injured retina. We're now exploring even longer time points. Obviously, many people are exploring doing this kind of experimental design in glaucoma models now as a different optic neuropathy. We did, however, again, when you think about changing the environment, we did notice in these experiments that merely doing the transplant actually saves the host cells from dying after optic nerve crush. And so, again, this premise that there is the opportunity to modify the environment through the transplant itself, I think is a really important one to think about. And then the other question that I just wanted to make sure that we seed here today is, in addition to these direct questions, is the premise that donor cell host environment optimization is still likely to need us to attend to the donor cells. In other words, there may be different optimal donor cells depending on these important questions like what was the optic neuropathy, or what is the disease severity or stage or time after transplant. And it may be a different optimal cell type that works best in any of these different conditions, if you will. And so we just have to, I think, be attentive to testing those kinds of questions directly. We have published some data, again, suggesting some elements that point to this. One is that, if our cells, the donor cells, had good survival in parallel cultures, we had many more successful transplants. If we had poor survival in parallel cultures, then we had poor survival in vivo. So again, optimizing the cell for the donor event. We also found, when thinking about the cell numbers, again, like at the time, it was paradoxical that fewer cells injected showed more cells retained. More cells injected actually showed fewer cells integrating into the retina. And again, one might reasonably hypothesize that this is, perhaps inducing more inflammation in the donor, in the recipient retina. So again, like attention to that post-environmental element. Interestingly, though, when we looked at the number of transplanted cells integrating into the retina, and compared that to the number of transplanted cells with neurites or with their neurites reaching the optic nerve head, these seem to be pretty linearly correlated. The more cells you can put in, the more cells that bear neurites and make it to the optic nerve head. So small proportions, of course, but still, there's going to be elements here. Maybe neurite growth is a little more independent. Maybe integration is a little more dependent on cell number or host factors. So I think these are experiments that are still ahead of us. I think that being really attentive to the maturation stage, not just the severity of the host environment disease, but also the maturation stage of your donor cells, and really studying these in head-to-head formats. Here, Ziming has shown that the 3D organoid RGCs are less mature than their 2D stem cell derived RGCs, both when looking at gene expression and also when looking at physiology, like measures of electrophysiologic differentiation. And again, these data are all published now. But I think, again, this points to the importance that we do some of these kinds of experiments really head to head, like across severities of disease, across donor cell types, etc. So I'll stop there as kind of an opportunity to have seeded some points, seed some points for ongoing discussion that I think we'll want to tackle as a field.
Wonderful, thank you. Let's turn over to Tom Johnson.
Thanks. 1 second here as I get the slides up. All right. Looking good?
Yep.
All right, great. So I'm going to follow on from that. I think fantastic points have been made so far. And one thing that I really like about this forum is we kind of can put on the table all the different key pieces of data and observations that have been made across labs so far and try to see where some of the commonalities and take-home messages might be. So when you think about the environment, both for how to optimize the survival of retinal ganglion cells that you're transplanting and how to get them to do what you want to, meaning sending dendrites into the inner plexiform layer, growing axons into the brain. There's a ton of things to think about. And I think in many ways, this parallels the 20 or 30-year history that glaucoma has in neuroprotection research. Anything that may have been relevant to the glaucoma disease process or other optic neuropathies and targets that have shown promise in reducing endogenous retinal ganglion cell death may in some ways be fair targets to try in conjunction with transplantation. I think one thing that we should not lose sight of is depending on how you transplant RGCs, they may encounter multiple environments. If we're injecting cells into the vitreous cavity, that's going to be a very unique environment as compared to what they're going to experience if and when they engraft into the retinal parenchyma. And some of the longitudinal imaging experiments that we've done in our lab suggest that the greatest drop-off in RGC number occurs in the first 24 to 48 hours after transplantation, which may be when the cells are trapped in the vitreous and may not have access to the oxygen and metabolic support that they need. So half the battle may just be getting the cells to a more supportive environment, which may be the retina, even though it may not be optimal. Lots of other things people have looked at in the past, macroglial reactivity, and I think that includes both the reactivity of Mueller glia and astrocytes in response to the disease that's going on, and then any intervention on the eye, introduction of new cells, exogenous materials is likely to further modify the phenotype of both the macroglia and the microglia. And then, we have a lot of seminal work from optic nerve and from spinal cord and other parts of the brain where we can point to inhibitory molecules and things that may be preventing the functional engraftment of these cells as much as they can, like CSPGs. I know Larry's on this call, so I have wondered myself what the role of free zinc may be in transplantation because it's starting to be better appreciated in disease states, and a couple of other thoughts on here. To make a few points and discuss areas for future work and collaboration here, we've been hypothesizing for a few years that the ILM is a barrier to retinal ganglion cell integration, and in light of the idea that cells being trapped in the vitreous may be more susceptible to early death than those that make it into the retina, this may be an even more important point. These are some of the first observations we made that if there are iatrogenic tears in the ILM, then donor RGCs labeled here in red can get into the retina and migrate laterally underneath the ILM. We've since developed methods for experimentally disrupting the ILM in rodents with proteolytic enzymes, as well as using transgenic mice that have developmental abnormalities and naturally occurring holes in the ILM. And we found that if you disrupt the ILM, not only does it improve retinal ganglion cell survival at early time points after transplantation, but it also improves engraftment. It increases the ability of somas to migrate underneath the ILM into the retinal parenchyma, and it also improves the ability of them to elaborate dendrites within the inner plexiform layer. And this here is essentially a quantification of what's shown more convincingly in this 3D rendering, where you have a retinal ganglion cell here in conjunction with three other cells that failed to engraft into the retina. But if we trace the dendrites of this cell, and look at it in 3 dimensions, it's clear that this cell body is coplanar with the endogenous RGCs and that the dendrites that are being laminated are within the inner plexiform layer. So, getting past the intrinsic barriers to engraftment are important. And we've recently now found that if you can get RGCs to laminate dendrites within the inner plexiform layer, they will form synapses to the afferent bipolar cells. We've done this using calcium imaging. So on the right is a field of transplanted retinal ganglion cells, and every 10 seconds you see them gain fluorescent intensity. So that's the GCAM molecule reporting an increase in electrophysiological activity, which is coincident with us shining visible light onto the photoreceptors of this retina. And again, here, every time there's a yellow line, that's a visible light stimulus. Endogenous RGCs have, as you'd expect, a very strong response to visible light. But donor RGCs that have successfully engrafted into the retina also have a response to light, whereas unengrafted cells do not. And importantly, if you block synaptic communication with cobalt chloride, that abolishes the light reactivity of these cells. And so we do believe that this is synapse mediated and not due to intrinsic photosensitivity. I don't have slides for this, but to discuss other points, we've done this in rhesus macaques now with ILM peeling in conjunction with pars plana vitrectomy, and have shown similar observations that if you peel half of the macula, the RGCs will survive better and they will structurally integrate into the part of the macula where the ILM has been peeled, but they tend to survive poorly and not integrate where the ILM is left intact. And then as a final point, just to add on to what Jeff was saying about transplanting after optic nerve crush, we've done this after both optic nerve crush and microbead-induced ocular hypertension as a model of glaucoma. And interestingly, our findings in optic nerve crush mirror very much what Jeff was saying. We've seen extremely poor survival of donor cells after optic nerve crush when we transplant at 1 week, 4 weeks, or even 12 weeks after injury. However, when we induce ocular hypertension and kill endogenous RGCs through an IOP-mediated mechanism, we actually see significantly better retinal ganglion cell survival and better engraftment. And so I think this suggests that the injury mechanism is very important and is probably leading to key changes in the microenvironment that these cells are experiencing that may suggest treating different optic neuropathies is going to require, as was suggested before, different unique approaches. So I will stop there and we can go on to our next speaker.
Great. We'll turn it over to Peter Baranov.
Thank you, Matt. Thanks to RReSTORe for this great opportunity to talk to you today. Let me start sharing soon. Okay. Yeah, and it's great to follow up on the presentations from Brad, Jeff, and Tom, and discuss what we are doing in the group and kind of what's the direction that we're exploring when it comes to making the optimal host microenvironment for retinal ganglion cell replacement. I think the challenge that everybody mentioned today is that adult retina and optic nerve, they are not pro-regenerative, and that's what the tremendous amount of work from Larry Banowitz and Jigan He and others in the regeneration field, not necessarily in cell transplantation, have shown that you need to do something in order to make retinal ganglion cells regenerate and grow. For us, when it comes to transplanting RGCs, we decided to focus on the recapitulation of the development as a roadmap, as a strategy to improve the transplantation outcome. And if you think about it, the cells have to go through several stages. They have to survive, they have to connect within the retina and the brain, and then later they have to mature molecularly, electrophysiologically, undergo myelination, and eventually acquire the function needed. How many cells, donor cells, we need to get at the top of this pyramid? As Jeff said, maybe a few thousand. But it definitely means we need to start with several, like tens of thousands, hundreds of thousands, because at each of this step, only few cells actually go to the next level. And so an argument that, to make an argument that development retina is a good microenvironment, if we deliver stem cell derived RGCs, and in this study done by Julia Oswald in the group, these were mouse-to-mouse transplants. So mouse stem cell-derived RGC differentiated in organoid system delivered into the developing eye. We can see a pretty nice integration here, donor cells in green, but that does not happen when we deliver stem cell-derived RGCs into the adult eye. I'm not in the same paper. Also explored the role of different mechanism of damage. We haven't done optic nerve crush, but we compared IOP elevation and NMDA toxicity. And in both of those scenarios, the survival was of donor RGCs was much higher compared to the adult retina, but it never approached, but it still remains in the fractions of percent. It never approaches the survival and integration that we can observe in the developing retina. So how to go about this and what are the factors that we need to add? And Tom summarized them nicely, all those interactions. And we're trying to cherry pick where do we start, and just go one by one. To do it, we put together developing an atlas from single cell data for developing RGCs. And it allows us to study these cells, the single cell level, see what does it take to make them from the retinal progenitors, and what are the changes they undergo, including primarily the receptors for the neurotrophins, for cytokines, for neurokines, so things that we can approach with some engineering solutions. And we also can study there are different tools available, and this is an example of studying this cell-cell interaction within the retina, within the developing retina. But you can, and we also have done this for the optic nerve crush for mouse data. But it would be really nice to explore this interaction at the high throughput level in other diseases as well, to approach the question that Brad posed, like, is there a good and optimal disease and the disease model to start this cell replacement, cell restoration journey? I also can also break this by function, and that's really how we do it in the lab, a little bit of reductionist approach, but it works. And we most recently we focused on migration survival with migration. Also based on the single cell RNA-seq data, we were able to identify several receptors on donor stem cell derived RGCs that we can attack with ligands. We tested them in vitro using chemokines, and our top performers here were SDF1 and netrin. And we also shown that, like, if you create a gradient of SDF1 by depositing SDF1 slow release into the vitreous and then deliver donor RGC in the subretinal space, that would cause donor RGC to migrate from the subretinal space into the ganglion cell layer. When it comes to migration, the tool that we use right now is co-treatment with BDNF and GDNF slow release system. It works quite well, both for mouse and human RGCs, and now we get like survival in the range of hundreds after injecting 10,000, 5,000 RGCs per mouse eye, and it really allows us, enables to do the quantification necessary to compare different kinds of treatments. And in terms of the immune response, another finding that Moni Che pioneered, that directional work in the lab, is looking how to block RGC, donor RGC host microglia interaction, either with an Axin-5, and later Olga Malechka in the lab showed you can also reprogram microglia with soluble Fas ligand, which would allow us to achieve high survival, and as you can see here, even exon ingrowth, donor RGC exon ingrowth into the optic nerve head. And these are xenotransplants, and with our work in multiple species, when it comes to both donor species and host species, the microglia response is really like the universal response that works across these barriers. And like we think this is something really to consider in transplantation. This has to be suppressed because it happens in the first few days. And if we don't do it, like most of the donor cells will be eliminated. Like a short summary about the field, I think it's really about quantification, and something I want to put up for discussion for this group is that we were very much inspired by the work from Tom Johnson when they looked at the quantification of neurites and cell bodies in the explant assay, and I think that this can be achieved with in vivo, and this is really what is needed in order to make this experiment synchronized and make it a joint effort. And another way to get there is to use advanced integrated transcriptomics and single-cell RNA-seq, which now becomes more affordable and accessible, because once we get the data, the way we can work with the data sets is experiment and control agnostic. So we can integrate these data sets and look at the donor cell response and host response across different conditions in order to come up with an optimal microenvironment. That's it.
Wonderful. Thank you. All right. I'd like to turn it over to Derek Wellsby for our kind of final introductory remarks, and then we'll open things up for a broader discussion.
So very glad to be invited here, and it's obviously great to see friends and colleagues on the call. I realize I'm going last. I don't actually do a lot of transplantation in my lab. We focus more on neuroprotection So I want to make this relatively quick because I realize we're more than halfway done with this session. We haven't even started the discussion. There's so many people here to learn from. So I'm just going to raise some like big ideas, which I think many have been raised. And that is, to keep these cells alive, what are the pathways we want to target? What are the specific targets that are, things we're going to want to inhibit and things we're going to want to activate? There is going to be, at least with some of these, this fundamental conflict between the ability to keep a cell alive and to be able to promote axon regeneration. Then there's the details of how we're actually going to do this. And of course, as Tom mentioned, the timing of how we're going to do this. The challenges are different early on versus late. In terms of pathways, I think many of these have been talked about. I think we should not discount the role of axon injury. And so axon injury signaling, we're going to have to dissociate these cells. They're going to be in the setting of a diseased optic nerve head. And so keeping that axon alive, keeping the cell alive despite the ongoing axon injury signaling, I think is important. Another concept that maybe hasn't been raised is the idea of how many of these targets that are oncogenic or tumor suppressors are fair game. If we feel like we're doing things in RGCs specifically, is that something that is, people are comfortable with? There are a list of specific topics. Obviously, our lab, Don's lab, we've done some work on these guys. Gain of function, I think I want to move relatively quickly. We've shown that DLK is a great target. Unbiased functional genomic screening going through every gene in the genome has shown that simultaneous inhibition of DLK and LZK gives you the most robust survival of a dissociated neonatal RGC. When we inhibit DLK, LZK, we get basically near perfect protection of RGCs in the optic nerve crush model. but it comes at a cost. And that cost, but we can do this in vitro, is when we look at gene expression. If you look at a retina where you've just done optic nerve crush and you look at total gene expression changes, you get something like 4,000 different differentially expressed genes. When you do this in the setting of some of these very profound neuroprotective interventions, you end up with very few changes. So not so much of the RGC injury signal comes through a few mediators. And the consequence of that is that when we inhibit DLK and LZK, we actually inhibit attempts to regenerate. So there's going to be a challenge for the field, which is, yeah, we can keep cells alive, but if you want them to regenerate their axon, they need to know that they're injured. And some of these very profound or very robust neuroprotective interventions are going to prevent that. We've tried to get around that and identify targets that allow for regeneration and survival. This identified this GCK4 kinase story, which I'm just going to skip forward so we can get to some discussion. But this is work with Anna Latorre that I want to show right here. And that is when she has done transplantation in the absence of some of these neuroprotective interventions, you see, some scattering of surviving RGCs. But when you do it with some of these robust kinase inhibitors that are preventing axon injury signaling and the death that you get from dissociation, you see much more survival. But again, we have not figured out how to get that survival and integration. So we can get things to survive in the vitreous, but we were not getting things to survive this way in the retina. And with that, I just want to quickly, highlight some ideas, but let's get to some discussion because I think we only have, 39 minutes left.
All right, thank you so much. I'd like to open it up to the group. I did see some comments and some discussion going on in the chat. Diane, I know that you had some items that you raised. Did you want to bring any of those up sort of for the entire group?
Yeah, I mean, there were some answers in there. One of the things that I was talking about was, with the whole pathfinding, right? Having to cross over the optic chiasm and what kind of hence Gretel breadcrumb trail are we going to have of positive and negative pathfinding? Do we have to like pre-prime the path? So that's one of them. And then another one I was thinking of is we're actually funding an Alzheimer's project with FNIH that's pre-competitive with a number of other pharma companies about using SV2A PET in humans to try and see if that biomarker can show that, active connections. So are you going to, try to make sure that even though the cells are connecting, it's actually active? So are we, using SV2A or something else? We thought about that. I'll just throw that out for discussion.
I agree. I've always been fascinated by understanding how much of the guidance cues are still there in adults versus development. So, but I know nothing about this field. So who does? Who do we have on the call?
From my neural development, angiogenesis, and zebrafish, field, right? So you're right. A lot of things all disappear during development, so you actually have to pre-paint it down, maybe how to do that. Anybody have ideas?
Well, I have one thought. I mean, Jeff, Mom, you've shown and others have shown in zebrafish, there's a ton of mistargeted axons at early stages. And if you wait long enough, it seems like they kind of eventually find out where they need to go and prune back misguided axons, right? And I'm wondering, there's a lot of people on this call have done a lot of the seminal work on optic nerve regeneration in mice after optic nerve crush. Has anyone ever really gone out like 6 months and looked at the brain and seen if the number of cells that reinnervate the central subcortical centers continues to build over time? If, there's maybe the pathfinding cues are not necessarily there, but given enough time and a little bit of the cue, they'll eventually find their way.
Hey, Tom, can I show a couple slides of some work that we did in the squirrel monkey because I think it's relevant to this issue?
Yeah, that'd be great.
All right. this will be quick, but it's published stuff, but I think it's good to just go over this in this context because I think it bears on this. So this was several years ago. We used squirrel monkeys for this work, and we were actually not trying to restore ganglion cells at all, but we were trying to restore photoreceptors. This is work funded by the Foundation Fighting Blindness. So we used a protocol at that time that Deepak Lamba and Anna Latorre also was working on this project to grow these cells. And it was not organoids, it was sort of like pre-organoids where they form rosettes, but then you just let them mature in culture from these rosettes and start growing photoreceptors and ganglion cells. So essentially, you never go through the organoid stage, you just go through the stage where they're making nice rosettes, and then you let them develop that way. So we did, this was the way we grew our stem cells back then. Deepak's protocol was basically one of the first for growing retinal cells from stem cells. And we harvested these guys after we labeled them with GFP. And what Jen Chow and Todd Klissert did was they made subretinal injections by going through the sclera, through the choroid and through the sclera, and then made subretinal injections. And when they used one of these eye science catheters that you could thread through the anterior eye and then bring it all the way to the back of the eye, and then you could literally like put the cells in the subretinal space this way, and we could watch the bleb forming, and it was, we could see that it was lightly GFP positive as we were putting these in. We put about 100 microliters of fluid in the subretinal space in these squirrel monkeys. Now, the eye of a squirrel monkey is a lot smaller than a macaque, and so that's something that I'll come back to in just a sec. But what happened was over time, 4 weeks, 8 weeks, 12 weeks, we followed this with a RET cam, and this was the original transplant. Here's the optic nerve head. Here was the transplant. It was close to the fovea, not exactly in the fovea, but you could see it's like a blob of cells sitting in the subretinal space. And over time, they start making axons, and you can see better here at 12 weeks. You can see the axons coming from the transplanted cells and heading towards the optic nerve head. So we thought this was quite remarkable that they're actually tracking on the existing axons and growing towards the optic nerve head. But I have to say, when we analyzed these animals later, this animal later for the histology through retinal sections, you can see axons going all over the place. Like they were going peripherally as well. Many of them did grow centrally towards the optic nerve head, but many of them grew all over the place. And yet the other really strange thing was, you can see here probably best in C, a lot of these axons, the transplant is out here. A lot of these axons are growing not through the ganglion cell fiber layer. They're growing to the optic nerve head, but they're growing through the inner plexiform layer. So I have no idea what that's about. I was thinking that they were all following fascicles, but when we did the histology, they're not. So they're getting to the optic nerve. Some of them are getting to the optic nerve head via a completely different route, but they do seem to be, the majority of the axons were growing in the right way. This is an adult squirrel monkey. And then we did further characterization of these just to make sure it wasn't material transfer or anything like that. We looked at the GFP, and then we used human-specific antibodies that would just stain for the axons that were also GFP labeled. So and the other thing that was quite clear was they're not growing the way normal axons are. These were axons growing through the parenchyma of the retina, but basically not in the optic nerve fiber layer, but through the plexiform layer. So I guess I'll just end there, but just to say, my point is just that, let's see if I can end this, is just that you may be able to, it may be that we're going to the right place, but not necessarily through the same route. And the other point I wanted to make is, when we followed up on some of these experiments using organoid-derived ganglion cells, we don't get nearly as much robust ganglion cell integration or axon growth. So there was something about that protocol where the cells were plated, and I think Jeff has noticed this as well, is that when he pre-plates the organoid-derived ganglion cells, they seem to have better axon growth when transplanted into mice. So I think there's a couple things that about the environment, and it may be species dependent, but it may be that it interacts with the way the ganglion cells are prepared. But I think we want a set of ganglion cells that'll be really robust, both in survival and axon growth. And you know, that may be that we have to tailor the donor to the environment. Again, reiterating some of what Jeff said, but I think in our experience that the two things may go hand in hand, and you may need to get a ganglion cell that's just like a super grower and a super survivor. And you know, we might be able to take some of the examples that people have done in various species so far, using, you know, looking at their gene expression, trying to see some common set of factors that will convey that. And so, yeah, I'll stop there.
One of the questions I see coming up in the chat is from Dan, is even in the case of a degenerated retina, are there still, you know, tracts that growing axons can follow their way along? And this might speak a little bit to kind of how bad the disease condition is. Does anyone have any thoughts or insight on that?
Well, I think this data, and I remember reading this paper years ago, but I had forgotten about the location of the axons within the tissue. I mean, I had assumed that the cells must have crawled up onto the retinal surface, and again, that they were following existing fascicles. But this suggests that that's not necessary. You know, somehow they knew where the optic nerve head was, even though they were crawling below the level of the axons. So
yeah, and
I'd
I
love to
think
know what
that's
the cue is.
yeah, I don't know how to think about that either. I was really, like I said, I was really surprised. We did find that some of them grew, like I said, peripherally and went all the way to the ora serrata of the retina from that transplant. And those seemed like they were more likely to be sitting in the optic nerve fiber layer. But this is a normal monkey too. So remember, it doesn't have degeneration of the existing ganglion cells. And so it could be there wasn't enough space in the optic, you know, in the optic fiber layer, because, you know, maybe the axons needed, you know, were trying to get there, but instead they were finding a different way. But one of the other points that people made, and I've been thinking about this a bit, is the idea that if we could just get the axons to fasciculate with the existing axons, if 80% of our visual system is midget ganglion cells, and of our axons are midget ganglion cells, and we know where they go, most of the LGN, so the odds are with us if we just got axons to fasciculate with other axons, just follow the train, you know, and you're going to most likely get to the LGN if we put in midget ganglion cells and we just make them follow other fascicles. So we may not have to engineer the whole pathway. We may be able to just, you know, hop on the road, as long as there's some left, and as long as all the midgets aren't gone and really end stage disease. That would be at least one possibility that I think we might take advantage of.
Dr. Benowitz, I saw that you had your hand up there.
I did, but I'm not sure my point was relevant. I was going to show a slide showing that adult retinal ganglion cells in a couple of studies have navigated their way back to appropriate targets. Here, just from our own work here. So I think this part of it is, you know, certainly true. We feel very confident about this, that with appropriate combinatorial therapies, this is inducing intraocular inflammation that elevates levels of particular chemokines from neutrophils and macrophages, combined with P10 deletion. We see this amazing synergy. So the injury site is over here where the asterisk is, and these are axons regenerating the full length of the optic nerve all the way down to the chiasm, some percentage cross. In this paper from 2012, we reported that some of the axons are pretty clever and find their way to the DLGN. This result was contested by Kevin Park, and there have been, you know, various studies that show a hint of this. Kevin's study, I think, was done well. But in any event, several people have shown innervation of the suprachiasmatic nucleus, including Kevin himself and Kai Liu. So yeah, the axons still retain some level of intelligence in guidance cues, but again, this isn't transplanted RGCs. These are the endogenous mature mouse RGCs finding their way home.
Yeah, I've always pointed to your work and Kevin's and Jeff and some others as being a really important proof of principle that regenerating the optic nerve with transplanted cells is feasible. But in addition to it being endogenous RGCs versus stem cell derived or transplanted
Right.
RGC, I guess the other fundamental difference is the axon is the stump that gets crushed or transected is already in the optic nerve when it has to get going. And
Right. And
so
not only
ours,
that,
we're going to have to navigate through the optic nerve head as well.
right. Oh, not only that, but we know that the endogenous RGCs have at least the chance of retaining topographic cues that should enable them to form a topographic map when they find targets. But we have no idea if that could be recapitulated by transplanted cells.
Right.
Pete Williams, I think I saw your hand up.
Oh yeah, I was just going to ask a dumb question, which is if axons are not very plastic, but dendrites are very plastic, well, why do we have to stick the cell somas so very far away? Can't we stick them in the optic nerve head or the early part of the optic nerve and make the dendrites go towards the bipolar cells instead? Is that not I think this is a dumb question that we can answer very quickly, but it's a thing going through my head watching Tom's talk going, yeah, axons find it hard, but if we stuck them in the optic nerve already, maybe they can just follow everybody.
I think it's a great question, Pete.
I mean, I think it's a bigger question as well is how do you get just this topography when you transplant? If you look at the exquisite organization of the midget circuit that people like Dennis Dacey have been reconstructing with pseudo block phase EM, it's quite remarkable how, essentially the ganglion cell dendrites are like a little private line where a single dendrite will have a synapse with two bipolars and then onto the cones. And so you literally, that kind of organization, as well as that topography, is what we need for really a great vision, kind of high acuity vision that we have. So I think there will be a lot of tricky engineering that will have to go into raying these cells in the appropriate way to get that kind of level of vision back. But I think, as Jeff said earlier, just getting no light perception to light perception, that's definitely a more achievable goal, and we probably don't have to worry too much about axon growth in appropriate topographical maps for that.
Don Zach, do you want to chime in?
Yeah, actually, I had two questions for the group. One is, single cell analysis have looked pretty nicely at the different subsets of ganglion cells or different expression patterns that survive better. But where do things stand with looking at single cell analysis of those cells that show the maximal axonal outgrowth for the cells that grow the most? Where does that stand? And number two is a question for Derek or anybody else. Looking at, related to the last question, where we put ganglion cells, how about relay neurons? Where does things stand now with putting in relay neurons somewhere in the optic nerve? So rather than get the axons to go all the way, find another cell, another neuron that can help with the process. So the two unrelated questions, but I'd be curious to see what the group thinks the state of the art is of those two different areas.
I can take the second one. And that is, obviously there's a lot of work that's been done in spinal cord, but what has always worked, and obviously, we've done work with Mark Tuszynski, Jean Doe in optic nerve. What worries me is that in spinal cord, you have these central pattern generators that you're trying to reinnervate, and your reinnervation needs to basically provide a binary on-off signal as opposed to the visual information that we need to send. So I think it's a much harder challenge in the visual system. That being said, people have certainly shown that visual information that's disorganized can sort of reorganize at some basic level. So we may not need a perfect conduction of signal. We just need to get something. But along that point, actually, to Pete's point, obviously a ganglion cell is the most reasonable place to start in terms of transplantation. But what about all the various other projection neurons? You know, could they have a role here if the job is sort of simple, just to get light, information back to the brain?
But if I might just interject, what will that vision be if you're introducing something that's physically different, like introducing an interneuron? Will we be able to, our brain be able to interpret vision in the same way?
And how much of that could be addressed computationally with VR and machine learning and
Yeah.
Also, how much of it is about restoring light perception versus trying to get really beautifully formed vision? And
Exactly.
again, these might be two different problems in the end to
baby
solve.
steps.
So I have a question on
Answer for Don's first question. I don't have an answer.
yeah, I have a question for both Don and Derek. This is with regards to the neuroprotection versus regeneration with DLK LCK knockout. Does it have to be knocked out permanently, or can you have a construct where you have suppression of DLK and LZK transiently for some period, initial period, and then it's restored in these transplanted cells?
Great question. So I know work that Tom and Don have done with Pingwu Zhang is, involves DLK, LZK knockouts. We're doing it genetically and it's permanent. But to your point, like you don't probably need it permanently, right? Once everything's reintegrated and getting the appropriate neurotrophic signals. That being said, I think still it's a challenge. For the period that which you have it inhibited, it prevents regeneration. And so I think there's still, even if you just do temporary DLK, LZK inhibition, I think there is still a challenge because you're going to need that axon to regrow.
And I think there's a second component, right? One is temporally timing, which is an important point, but the other thing is how much inhibition do you need? Can we titrate the amount of inhibition such that to favor survival partially and not inhibit regeneration too much? So, like everything in life, it's not black and white. So I think that's an area that obviously adds complexity, but it's important. And with CRISPR-A, CRISPR-I, other methods, it's becoming doable to start to do titrations.
And Trent Watkins is actually working on that very idea, actually using DLK activation at some low level as a way of getting sort of, not the death, but getting axon regeneration.
Or maybe some kind of pulsatile changes or something, whatever. Yeah.
Nick, I see that you have your hand up there.
Yes, just to respond to Don's first question about the regeneration across different cell types. We've certainly observed type-specific axon regeneration in most of the interventions that we've looked at. Certainly in P10, the alpha cells have the strongest growth potential, but that's not always the case. So, SOX11 is another treatment where if you overexpress it, you actually kill all the alpha cells and promote regeneration of an unidentified, other subtype of RGC. So I think, based on what you're using to promote regeneration, you can expect to have different responses from different ganglion cell populations. And of course, all of the work that we've done so far has been in mouse. So we still don't know how that's going to translate into, how different human RGC types are being able to regenerate as well.
I'd like to make a follow-on to that and something that we've discussed a little bit in other meetings, and that is subtypes of ganglion cells. And, we've often had in other discussions we'll say, oh, well, let's just get some ganglion cells first, and then we'll figure out the subtype later if we could just get any old ganglion cell to do something. But I'm wondering if might not be a better strategy to actually figure out, which are the ones that we would want to have, probably the midgets. What are the characteristics of those, and focus on stimulating the regeneration of axons of specific subclass of ganglion cells, because they're going to differ, as Nick's work has really shown. And so maybe if we just agree, at least pick a few that the glaucoma experts say those are the ones that are primarily lost in glaucoma, or these are the ones that really impact vision the most. And then we focus on that group, both for using stem cells to derive those specific subtypes, which I think we're probably getting better at, and or at least select them and have selection markers for specific subtypes of ganglion cells so that we can pull those out of the mix. And then specifically do some experiments designed for their survival, their integration, and their axon growth. So I know it sounds like that's moving ahead a few steps, but I think that's where we know we all want to go eventually. So given the variability in ganglion cells and their responses to these things, maybe it's time to start thinking that way.
Yeah, I mean, that seems like a question of what's going to be the biggest bang for the buck, right? In sort of where can the efforts be pushed that's going to give the biggest return on actual vision restoration. Yeah.
And then, of course, in human stem cell derived ganglion cells, we still really can't carefully make the specific subsets. We can have some heterogeneity. So, A, we want to figure out what we want, but B, we would need to figure out how to get the cells to fit those characteristics.
I have a question. Have we, oh, go ahead.
No, that's right. Go ahead, Diane.
No, I was just going to say, have we figured out which combination of RGCs or which RGC the best? And then how do we choose that from the patient? And do we have to do, if it's patient autologous, do we need to do genetic correction? Or is that on the list?
One of the discussions we had at the in-person meeting, Diane, was IP RGCs, because then you need a little bit less connectivity, a single target. And if we're talking about what might be viable for, a phase 1, phase 2 FDA approved trial, light perception might be something, because then we're working with already, completely blind, no light perception patients. And that's probably our lowest risk of entry. I'm not the expert here, but this is part of the discussion that we had last time. I don't know if anybody wants to chime in with more thoughts on that since we last discussed.
Had the discussion gone towards a certain type of RGC, or does it not matter?
Yeah, the intrinsically photoreceptive RGC is part of what was discussed, but anybody chime in.
Yeah, I just wanted to mention that when we were taking our keratocytes from human donor eyes and reprogramming them in the lab, we generally get more alpha, like expressing alpha RGC markers versus IP RGCs, right? So we don't get those markers, obviously, in our ROs from the glaucoma ones or the non-glaucoma ones. But what we are seeing is more expression of the alpha RGCs in ROs that are derived from the glaucoma patient ones. And then that, just to add to that comment, so when we do the ex vivo human eye perfusion model in the lab, we've just submitted our paper as well for that, is what we notice is when exotomy is already started as soon as we get the human postmortem eyes, right? So the gliosis, all of that stuff is there. But when we transplant the stem cell derived RGCs in the human eye model, it's all relevant to the pressure also, right? So when there's elevated pressure, there's less survival of these RGCs too. So I think there is also not only the severity of the disease, but how we are managing the pressure paradigm when we're doing the transplantation as well. So I think our preliminary work, obviously we've not done any outgrowth studies or anything, but just the survival of the cells in our ex vivo human models is much worse when there's high IOP compared to normal IOP, which would be kind of expected with the microenvironment. But when we put in a trophic factor in the system, in the same model, it's better survival under high IOP conditions.
If I could just add a comment on this topic, the Chiodo paper, which is indeed online ahead of print, and it's linked in the chat, so anyone can have a look at it now. I think there's a couple of important take-home messages from it, germane to this last part of the discussion. One is that many of the guidance cues for the RGCs are still present along the pathway, but the receptors may not all be there, or at least in all the RGCs. And so manipulating both the donors and the recipients, if we're really going to try to think about getting them to vision-forming nuclei instead of, restoring a blind patient's circadian rhythm. And the other point is that even when doing that, there was not particularly cell type specificity innervating subcortical or subthalamic even targets in that paper. And so again, it really may be like, hey, let's take any RGC subtype, but give it the tools it needs to get to the LGN, rather than worrying about generating a specific RGC subtype and hoping it's sort of reborn with those correct tools to get to the LGN. So I think a couple of sort of important initial data points in that paper linked in the chat.
Josh, did you want to chime in?
Yeah, the paper linked to the chat might change my opinion on this, but I was just going to say, all else being equal, if you want to save yourself problems downstream, you want the RGCs to be behaving like RGCs, and you want them to be midgets or alphas or whatever. If you want to restore conscious vision, and you hope that the thalamocortical cells don't degenerate and they actually respond to them and form synapses and everything. I think if alpha RGCs were a particularly difficult cell type to save or preserve or work with there might be a good argument against looking at an alternative. But if we're using mouse models and alpha RGCs are working reasonably well, and that's homologous to what we ultimately want to get to, I think that's a good way to go. If that wasn't too sideways and confusing.
Another aspect for those of us doing RGC transplants, it was mentioned by several people today that we transplant thousands and thousands of cells, and then a couple weeks later we see best case scenario a few hundred surviving. It's kind of reminiscent of what happens after optic nerve crush. It took a while for us to step back and say, oh, I wonder what's going on with these 10% of cells that are surviving. But thanks to Nick and some others, we now know that that's actually, that was kind of an intrinsic biological experiment that taught us a lot. And maybe we should be doing more work to carefully assay what's special about the 1% of RGCs that we do see surviving. Peter, I know you've been doing some single cell sequencing by pulling the RGCs back out after transplantation. How's that going? And does anybody else have any thoughts for trying to figure out what's unique about this very tiny population that across experiments seems to do better than the 99% of the other cells?
Yeah, I think thanks, Tom, for bringing it up. I think it's a great question. Like, is it nature versus nurture? Is it purely stochastic process that we get like that 10%, and can we increase the number just by putting less? The latter is definitely the case. We can increase the number by putting less, and that's why we currently transplant less cells. In terms of analyzing donor cells after transplantation, I think from the data, it's not, I cannot say it's final, but the preliminary analysis suggests that like the cells that eventually going to make it, they are different to begin with. So they're less stressed, and that's kind of goes along with what Jeff brought up years ago about doing parallel cultures. So like plating cells, they can use in survival after plating new right outgrowth as some sort of quality control for your transplants. So I'll just leave it here for the sake of time, but happy to talk more.
How much of that is because of shear stress during injection?
Good amount, and that's why another trick to use for those transplanting is like put your cells in hydrogel. I don't know, Tom and Jeff actually used some of the either hyaluronan or something else for the other formulation for RGC transplantation. We use just basic hyaluronic acid now. I mean, all the data I've showed is without any gel, but like we use like low concentration hyaluronic acid just to prevent the shear stress. And that's why, Kellen, back to your point, if for those quality control studies, when we plate cells back, actually these are the cells that go through the same, 38 gauge needle or glass needle that is used for the injections.
That point seems a little counterintuitive to me, Peter, because the increased viscosity would make for higher shear force. Maybe not directly on the cell membrane, but generally just physically within the bore of the needle,
It may to do good. We haven't investigated that on that level, but it may to do with cell clumping as well. So if you put cells in a more viscous, slightly viscous liquid, it does prevent them from clumping, and then they would go smoothly.
Meredith, I see your hand up. You want to chime in?
Yeah, so just, I mean, to kind of, I'm not, I've been doing some work with Peter on kind of modifying the, the host environment or specifically the microglia. And so, kind of looking at that too, in terms of, looking at Larry Benowitz's data that he just, that he showed, and even some of Dongfen Chen's work, looking at, how the role of glia and inflammation can actually also work towards stimulating axon regeneration, and kind of not looking at this in a vacuum just to get the ideal ganglion cell, but, the ideal ganglion cell with maybe the ideal microenvironment, or what factors these glia are producing that, are beneficial, enhancing axon regeneration versus those that are inhibiting it. And is there some way to, in high throughput, looking at that, in vitro, ex vivo?
I mean, Peter's data speaks a little bit to that, Meredith. The using the annexin-5 as to mask an eat me signal, so to speak.
Right.
We had some other plans as well to manipulate the host retina, suppressing using a CSF1 inhibitor, not a Plexicon. Initially, that was our idea, to use the Plexicon drug, but it turns out to be too expensive in monkeys for the dose you would need to achieve, at least if we make the parallel to mouse.
Well, but I don't know that, like some of Dongfen Chen's work, I mean, and other work out there that, I don't know that you want to, I mean, I'm pretty confident you don't want to eliminate.
No, it's a good point.
Yeah.
Good point.
I mean,
actually,
modifying it, right?
Serendipitously, Yvonne O suggested to use, there's NLY-01 as an inhibitor rather than, make a manipulation to suppress microglia and not eliminate them, not deplete them from the retina. So that was actually our follow-up plan,
right?
Which we haven't implemented just yet.
Right. I mean, that's, the work that I'm doing with Peter with the fast ligand too, is this idea of kind of resetting microglia to maybe more of their homeostatic phenotype, and maybe more supportive of the ganglion cells. But
so Jeff was alluding to that by saying just wait. If you wait long enough, microglia may do that on their own. I can say that we haven't had dramatic success in very sort of end stage candidates that had glaucoma for such a long time that maybe things had gone quiescent by then. It may be very different for a crush, or transection type approach than
Right.
it would be for glaucoma.
Well, that's something that, the data that some of you guys were showing today, looking at the different models, like, whether you have, more success, more ganglion cell survival, integration, depending on the model. I mean, what are the differences in the glia or the microenvironment in those models? I mean, is it the severity of the injury, or are there differences in the environment? Right.
Like the synchronicity, from a crush, it's everything's happening all at once.
Right.
Um,
Right.
I think we are getting close to the end. So I see Nick, your hand is raised. Did you have another comment you want to add or a question you want to ask for the group? And then shortly after that, I think we'll close our session.
Yeah, so this is a question for maybe for Peter or Tom. So, in the peripheral nervous system, like preconditioning lesions have been really shown to, improve the response to like a second lesion. So, you'll get more robust regeneration and neural repair. Has that ever been tried in this system? And you say like the transplantation, you mentioned that there's a lot of shearing, there's a lot of damage. Of course, you're lifting it off the plate, but have you guys ever tried like replating them first, making essentially have like one shearing lesion and then putting them back in and then seeing if they'll have better regeneration after that?
Yeah, we haven't tried that. I think it was alluded to that Jeff has been plating organoid-derived cells. So that's sort of like that, but we haven't tried from a 2D differentiation, detaching them, replating them, and then detaching them a second time. It's a really good idea. I mean, we've also thought about like hypoxic preconditioning, given that they're going into the vitreous. I think that there are probably several preconditioning paradigms that could be explored. I think it's a good idea.
Yeah, and Larry may comment on this too, but classically, preconditioning lesions and optic nerve regeneration in goldfish that was one of the first areas where people had demonstrated this. And so, yeah, for sure, it does work in other vertebrates for ganglion cell axon regrowth.
Yeah.
I don't know, Larry may also remember some
Yeah,
of those old studies.
Yeah, we tried it, Tom, exactly for that reason. It didn't seem to work. It works great in peripheral nerve, and that's related to inflammation, and maybe the goldfishes, but we didn't see it in mice.
Yeah, so we do that. We actually dissociate the ROs and then plate them, and then we culture them for at least 10 days with trophic factors, and then we do the transplantation for the ex vivo. So I think that really works better rather than just doing the dissociation and then putting it back in. So I think just replating them, putting them in 2D culture, and then putting them back, that really helps them survive better, at least for us.
Compared them head to head though?
Yeah. Tom, sorry, I didn't hear you.
Oh, I was just asking, have you compared that head to head with acute dissociation and transplantation?
Yes, with acute dissociation, they just die much faster. So when we do just the acute dissociation, they just die much faster. Because I think they need to have that sphenoid outgrowths, and then we just plate them off, and we do either Versine or the space, and then just lift them off, and then just do the transplantation. So that works a lot better.
Tasneem, can I ask, please, where are you doing the transplantation in the
ex vivo
cultured eye
human
model?
Yeah,
but
we
where in the retina? Where in
we
the eye?
do it in the middle, so around the optic nerve head area. So that's where the problem is. We don't do it all around the whole retina, but just
Subretinally, intraretinally, in the vitreous, where are you putting cells?
it would be called intravitreal, but the eye is cut in half. So we just do a posterior dissection.
Oh, it's just the posterior eye cup.
Posterior
Gotcha.
eye cup. Right.
Gotcha.
Yeah.
And when you mentioned that when you pressurize that eye cup, you get worse survival unless you have some growth factors. When you say pressurize, how are you pressurizing an eye cup without affecting the perfusate?
Yeah, so we basically have an eye model system in the lab where we cut it in half, and then we take the whole posterior segment and put it in there, and then it seals, and we seal it and pressurize it, so we have normal pressures and high pressures, and there's always a contralateral eye. I know we're running out of time, otherwise I could show like a quick diagram of what we have, And if everybody doesn't mind, I could. It would just take 30 seconds, but I could just share it just for relevance purposes and
I just
reference.
wanted the quick question of, like, you're maintaining flow of the perfusate despite having high pressure. Yeah.
Yeah, so that's kind of how we have it. And so that's how the system is. So then we can basically, we cut the eye up and then the eye just goes onto the model system. And so when we are even doing the transplantation and we keep, once the vitreous is removed, obviously it doesn't hold the retina in place. So then we have a contact lens to keep it in place. And then we'll just, but for when we do the transplantation, we keep it 24 hours in the posterior eye segment before we do the perfusion.
Gotcha.
And then we can have it at different kind of pressure conditions. So we can go all the way to 40 millimeters of mercury if we wanted to with the human eye model system. So, but this is kind of like what I was talking about when I was mentioning that when we try to do it, so we grow them in culture and then GFP tag them so we can kind of, the recipient and the host, we can kind of distinguish it. And so when we keep them under normal or high pressure, the high pressures are around anything over 21 millimeters of mercury. But as you can see, there's more cells that are within the normal IOP versus in the high IOP. Even the resident retinal ganglion cells are pretty much dying by that. And this is at 7 days after perfusion. So we see a lot more cell death after the transplantation too, and less survival basically. And so I'm going to stop sharing. I know we are running out of time, but
I find that quite perplexing. So
yeah, so we maintain the pressures. It's just that we always normalize it. So we are normalizing it to multiple things, right? We're normalizing it to what the basic cell death would be after exotomy. We're normalizing it to the contralateral eye that would not get the transplantation. We're normalizing it to the normal pressure versus the high pressure. So we tease out all of that before we get the survival. So we are kind of trying to tease out all those different layers of complexity that you would have.
it's a cool system.
That is the end of our time, so I think I'm going to call the session here. Thank you all for participating. If you have ideas or ways that we can improve, I'm thinking of putting together a survey for the entire group, and you can share feedback that way, although you're also welcome to directly email me if you have input or ideas for future virtual discussions. So I'd appreciate receiving those emails, and ideas are always welcome. Thank you all for your time, and thank you all for a really good discussion.
Bye, everyone.
Thank you.
Happy holidays. Bye.